When we think about how life started, it's tempting to look for a single magical spark. But the truth is a lot slower and a lot more physical. The origin of life was a long, gradual climb from fizzing chemicals and rocks to biological cells. Our very oldest ancestor didn't look like a complex cell at all. It was a protocell. You can think of it as a microscopic, simple soap bubble made entirely out of fatty acids. Modern cells have strict doors and heavily guarded walls to control what goes in and out. But these early bubbles were extremely leaky. They didn't even have to hunt or eat in the traditional sense. Food and nutrients simply drifted right through their soapy skin. At this point, the bubble wasn't actually acting biologically. It was really just a physical container floating in the ocean, waiting for a set of instructions. So how did these microscopic bubbles grow? Scientists call the process survival of the fattest. And it was driven entirely by basic thermodynamics rather than any kind of biological hunger. If a protocell had more genetic material trapped inside it, that material created outward tension. To relieve that pressure, the bubble physically stole fatty acids from its weaker, empty neighbors, expanding its own outer skin in the process. Inside that expanding bubble, the whole show was run by a molecule called RNA. Today, our cells divide the labor. DNA holds the information and proteins do the work. But back then, RNA did both. It stored the genetic data and it acted as a physical machine, called a ribozyme, that stitches things together. Imagine a recipe book that can literally cook its own dinner. Once you combine that growing physical container with a working instruction manual inside it, you establish the foundation. This is where chemical reactions finally cross over into biological evolution. Over a massive span of time, those protocells evolved into something much more sophisticated, LUCA, or the last universal common ancestor. LUCA was a complex, heat-loving organism that lived its entire life tucked inside deep-sea hydrothermal vents at the bottom of the ocean. LUCA didn't hunt for food. It used a chemical process called the Wood-Lungdahl pathway. Through this pathway, LUCA essentially breathed in hydrogen gas and carbon dioxide directly from the vent, turning those raw gases into energy and body parts. To power all this, LUCA plugged right into the planet itself. The early ocean was highly acidic, full of positive charges, while the fluid inside the vent was alkaline. When those two waters met at the rocky chimney wall, they created a sharp chemical contrast. That contrast created a proton gradient. Because nature wants to balance things out, the acidic particles, or protons, desperately want to flow into the alkaline space. It acts exactly like a natural, rushing waterfall of energy. Life built a microscopic motor in the wall of the cell, called ATP synthase. As those protons flowed through the motor, it started spinning, generating the cellular electricity LUCA needed to survive. We actually still use this exact same motor in our own bodies today. As complex as LUCA was, it had a strict limit. It was entirely dependent on that specific geological environment. It couldn't leave, because it was physically tethered to the vent's natural power grid. This brings up an interesting problem. How do we draw the line between a complex rock and a living cell? Scientists use a checklist called the chematon model to officially define what counts as alive. To pass the test, a thing needs 3 systems working together. It needs an engine to make energy, called metabolism. It needs a manual to copy itself, called a genome. And it needs a solid boundary or wall to keep it all contained together, the membrane. Early on, organisms that met these criteria were incredibly cooperative. Evolution was essentially chemical communism. Instead of competing, these simple cells constantly swapped helpful genetic tools and secrets with their neighbors, a process called horizontal gene transfer. Eventually, the cells became highly complex. If you randomly insert new code into a sophisticated machine, you'll probably break it. To survive, cells had to stop sharing random code. They locked their doors to protect their own specific lineages, crossing what scientists call the Darwinian threshold. Ending that friendly, cooperative web was a necessary sacrifice. Locking down their code is what allowed distinct species to finally form, officially sprouting the branches of the tree of life. As those branches grew, the cells ran into a major hardware limit. That original RNA manual is incredibly fragile. It tears easily, which means it physically cannot hold the massive lists of instructions required to build a larger, more complex cell. To get bigger, life needed a software update, and it found one in DNA. DNA is highly stable. It is a tightly bound, heavy-duty format capable of securely holding vast amounts of genetic data without falling apart. To make the switch, a specific enzyme acted as a cellular scribe. This enzyme, called reverse transcriptase, which life actually might have borrowed from a virus, carefully copied all the old RNA notes over into the new, durable DNA format. Securing a long-term, reliable way to store information was an absolute prerequisite. Without that stable genetic code, life could never have built the complex, resilient organisms we see today. But even with this pristine new DNA, our cellular ancestors were still physically stuck at those hydrothermal vents because they couldn't generate their own independent power. The breakthrough came with the invention of antiporters, specifically something called the MRP pump. This was a microscopic mechanism that allowed the cell to pump particles back and forth across its own membrane, creating its own internal power gradients. With this tiny pump, cells could finally pack up their own portable, rechargeable batteries. They were no longer tethered to the ocean floor and could freely travel out into the open water. As they migrated into the harsh open ocean, they realized their leaky, soapy bubbles wouldn't survive. They needed tougher outer skins. But as they solved this problem, the lineage split, using two completely different chemistries to build their new armor. This split is known as the lipid divide. One group became the bacteria, securing their outer walls with fatty acids called ester bonds. The other group became the archaea, using a totally different chemical link called ether bonds. Those two groups separated forever, forming the two great domains of life. The journey from a fizzing ocean to a free-floating cell was a massive, millions of years grind. It was driven first by the physics of bubbles, then guided by the chemistry of RNA, powered by the geology of deep-sea vents, and ultimately set free by biology, allowing our tiny ancestors to finally leave home and conquer the globe.